Hello everyone, my name is Tomasz Klaus. I'm head of antibody discovery team at Pure Biologics. And a few months ago, we initiated partnering with Twist Bioscience, and we currently use libraries from them. Today I would like to present data about performance of new library from Twist. It's a Hyperimmune scFv library. And let me introduce shortly Pure Biologics. We have been on the market for 11 years. Almost 100 specialists work at Pure Biologics currently. Our projects are based on proprietary technologies of antibody and aptamer discovery. And we work on the development of first-in-class new drugs and new therapies. Our focus is immuno-oncology and plasmapheresis. And the projects at Pure Biologics are based on two concepts, on antibodies and on aptamers. Antibodies, they do not to be further introduced. Aptamers are small DNA-based binding molecules, and they could be considered as a novel approach to specificity. In our pipeline, we have 5 projects. In 3 of them, we develop novel molecules for immuno-oncology. The 2 projects from plasmapheresis from aptamer stream aim to develop a device for plasmapheresis. Our projects are reaching preclinical phase. And so today, of course, I will focus on antibody discovery projects, which are at different stage and which share many similarities. In this immuno-oncology projects, we develop bispecifics. We work on immunoligands as well as immune cell engagers. As I explained, I will show you performance of Hyperimmune scFv library. Therefore, I will focus on the binding domains. The variable domains from the library will be incorporated into the chosen scaffolds and then further validated in functional assays. And a few words about the library. The Hyperimmune scFv library mimics the human antibody repertoire. It is based on common stable antibody scaffold. The CDRs from B cells were shuffled into this framework, and this library design removes common liabilities such as unwanted glycosylation sites, deamination sites, or hydrolysis sites. The overall diversity of this library is more than 10 billions of unique sequences. And here I prepared a kind of short summary of panning and library using for newbies in the field. Library is a collection of phages. Each of these phages displays on its surface unique binding domain. In the same time, DNA encoding this binding domain is inside the phage. Therefore, the phenotype is linked to the genotype. This collection of phages is contacted with immobilized antigen. Unbound phages are washed out, only the bound phages remains. These bound phages are eluted, for instance, by pH shift and then amplified. The amplified phages are subjected to another round of panning. After a few rounds of panning, the phages are sequenced to reveal unique binding sequences. And the word panning is derived from gold mining in a river sand. Phage libraries resemble sand with a few gold particles that are separated from sand by washing with water. And this is our workflow at how we generate antibodies at Pure Biologics. We do pannings on cells as well as on purified immobilized antigens. We contact these antigens with a library. Then we do a few rounds of panning and we analyze the retrieved pool in a binding assay. If we confirm an enrichment in this binding assay, we proceed with screening. We usually do screening in ELISA or in flow cytometry. After that, all identified hits are sequenced and the unique binders are validated in flow cytometry. At Pure, we work with our in-house libraries, therefore the implementation of libraries from Twist was quite easy for us, because we simply swapped our library with the library from Twist and we proceed with our standard operating procedures that were only slightly adjusted to the recommendations received from Twist. And a quick overview what we have done with the Hyperimmune scFv library. We did pannings on 3 antigens, it was TIM3 and immune checkpoint, it was ROR1 and the complex of GARP plus latent TGF-beta. We used different panning strategies. We worked on immobilized protein, on cells, as well as combined approaches were applied as well. And so far we have identified 35 unique binders. We still continue the screenings, therefore I expect this number will increase. And now I would like to show you the results from the TIM3 target. In the case of TIM3, our aim is to obtain an antibody that blocks TIM3 signaling. TIM3 binds its ligands via N-terminal domain, therefore this domain was our target. Moreover, we need an antibody that recognizes both human and cyno orthologs of TIM3, and to obtain the desired antibody, we must have prepared the antigens. We developed cell lines expressing N-terminal domain of TIM3. We produced extracellular domain of TIM3 in the recombinant form, and we purchased cyno antigen. We did 2 panning approaches. In the first one, we alternated cells expressing the N-terminal domain of human TIM3 and cyno antigen between the runs. In the second approach, again, we alternated human and cyno antigen, but we worked on recombinant proteins only. Here we have bar charts presenting screening results. From both pannings on recombinant protein as well as on cells, we obtained a lot of hits recognizing TIM3 as shown in green bars. We sequenced all of the identified hits, and from the panning on recombinant protein, we obtained 7 unique sequences, and from panning on cells, we got only 1. All these binders passed validation in flow cytometry, and we still continue the screening to obtain more of the unique sequences. To validate the unique binders, we usually apply flow cytometry. In the case of TIM3, we used 3 cell lines: wild-type cells and cells expressing N-terminal domain of TIM3. Here we have 2 cell lines expressing the N-terminal domain, because we used 2 different anchoring domains for this antigen. The cell lines were loaded with a different amount of green fluorescent dye, therefore we were able to identify them in green fluorescent channel. The mixed cells were contacted with phages. We detected the bound phages using red fluorescence antibody. And here I show the real results. On the left side, I present weakly binding non-specific phage that serve us as a negative control, and on the right side, there is a result for anti-TIM specific phage that is derived from our internal library, and we use this phage as a positive control here. So far we have validated 14 anti-TIM3 binders derived from the Twist library. All of them showed specific binding to the cells expressing TIM3. The identified antibodies recognize both human and cyno orthologs of TIM3. In overall, we have obtained 14 unique antibodies binding to TIM3 and terminal domain. and the antibodies are cross-reactive to human and cyno antigen. Therefore, using Twist library, we were able to obtain antibodies that met the selection criteria. And right now, let's move to another antigen, to ROR1. ROR1 is an antigen that is abundantly expressed during embryonic development. In adults, its expression is very limited. However, ROR1 reappears on malignant cells, and it could be used as a cancer marker. At Pure Biologics, we develop an immune cell engager, and this engager comprises two binding domains. Blue domain recognizes ROR1, and the green domain binds to a receptor on immune cells. This engager should attract immune cells to the malignancy expressing ROR1, and in the same time, this engager should activate cytotoxic functions of the immune cells. In the case of ROR1, we again did two pannings. The first panning was done on recombinant proteins, the second on cells. In both panning arms, we used for depletion step ROR2, and ROR2 is protein similar to ROR1 from the same family. And these charts present screening results. Green bars show binding to ROR1, whereas the violet bars show binding to ROR2. As you can see, again, we identified a lot of hits from both panning arms and we sequenced all of them. From the panning on cells, we obtained 14 unique sequences. From the panning on recombinant protein, we got 3, and one sequence was common for both panning arms. All the identified sequences passed validation in flow cytometry. So far, we have validated 18 anti-ROR1 binders. All of them, of course, are derived from Twist library. We still continue the screening, and I expect increase in this number. The ROR1 antibodies bind purified ROR1 from 2 different vendors and specifically bind to the cells expressing ROR1, binding to ROR2 that was used for depletion step was either not detected or very weak. And the third antigen is a bit more challenging. It's a complex of GARP and latent TGF-beta. This complex regulates activation of TGF-beta. Some of integrins bind the complex and induce activation or release of active mature TGF-beta. Our goal is to obtain an antibody preventing TGF-beta activation. We did panning on cells expressing the complex and of GARP and TGF-beta, and for depletion step we used wild-type parental cell line. After panning, we analyzed the pool of retrieved phages in flow cytometry. On the upper panel, I showed the controls. We worked with cell lines expressing GARP only, here in dark green, and the complex of GARP plus TGF-beta here marked in light green. The pool obtained after panning on the cells expressing the complex was analyzed in the same assay, and this pool was enriched in binders recognizing the cells expressing GARP and the complex. Moreover, the same pool was analyzed in polyclonal phage ELISA, and it showed binding to the antigens present on the target cells. And based on this result, we proceeded with the screening. And again, using the barcoded cells, we screened in flow cytometry around 200 colonies picked from the pool. So far, we have identified 149 hits, and we have just sent them for sequencing. On the slide, I showed 8 exemplary hits. We obtained hits binding only to the cells expressing the complex, as well as hits binding to both cells expressing GOP and the complex. Here I summarized the screening, the heat map, presence, relative binding to the target and the control cells. As you can see, a lot of clones bind the target cells specifically. We are going to sequence all the identified hits and reveal unique sequences of the obtained antibodies. And now it's time for a short summary. We pumped the Hyperimmune scFv library on 3 antigens, with it panning on cells as well as on immobilized recombinant proteins. For Team 3, we have identified 17 unique sequences. For Row 1, it was 18. Currently, we still continue the screening and we hope to obtain more. In the case of the complex of GARP plus TGF-beta, we send the clonal samples for sequencing. And based on our results, parallel panning on cells and on purified antigen is beneficial because the 2 panning approaches increase the overall number of unique binders. At Pure Biologics, we smoothly implemented the library from Twist. For us, it's an excellent tool to expand the diversity of molecules supplying our immuno-oncology projects. Randomization of Twist library and our in-house libraries is different, therefore we are able to obtain a broader range of molecules for functional assays. And I would like to say a warm thank you to all the people behind my slides. I combined here pictures of my beloved antibody team and few photos from our laboratory. And on the right side, I merged our cell biology team who helped us a lot with cells and flow cytometry. And last few words about Pure. We offer proprietary platforms for discovery of antibodies and aptamers. We have our own supporting facilities for protein production, for molecular and cell biology experiments, as well as for biophysical analysis. On board, we have more than 100 experienced staff, and we have been for 11 years on market. And thank you for your attention. Thank you very much for the invitation to this virtual event. And please find here our contact data.